Welcome, future toppers. Today we are cracking a topic that is guaranteed to show up on your exams: DNA mutations. These tiny little glitches in our genetic code are an absolute favorite of examiners, and really understanding them is the key to unlocking some serious marks. So stick with me to the end, because I'm not just going to explain the concepts, I'm also going to share some tricks and highlight the common mistakes that could totally cost you your rank. Let's dive in. Just think about that for a second. One single letter out of 3 billion gets changed. I mean, it seems so tiny, right? But this one idea is the foundation for questions on genetic disorders, on evolution, and even on cancer. Understanding the impact of that one little typo is what separates a good score from a great one. The stakes are really high here, so let's get this mastered. So to make this all real and not just theory, we're going to use the classic 5-star, super important example that you absolutely must know: sickle cell anemia. And why this one? Well, because it is the perfect model answer for any question about single gene disorders. It shows exactly how one tiny change in the gene that builds hemoglobin, the protein that carries oxygen in your blood, can lead to a massive, really complex disease. We'll keep coming back to this example, so lock it in. Okay, let's zoom all the way in. We're going to start at the smallest possible scale: a change happening in just a single gene. This is what we call a gene mutation, and it's pretty much where the story of most genetic disorders begins. Now, I need you to pay close attention right here. It is so critical to tell these two apart, because this is a really common trap on exams. You've got gene mutations, which are like single typos. And then you have chromosomal mutations, which are more like accidentally deleting or rearranging whole chapters of the DNA instruction manual. Seriously, students mix these up all the time, so getting this straight in your head right now gives you a massive advantage. So the most fundamental type of gene mutation is called the point mutation. And just like the slide says, this is a change that affects a single nucleotide base. You've got to remember that term, nucleotide, because it's the absolute smallest tweak you can make to the genetic code, and examiners expect you to know it. These single-letter changes can happen in one of 3 ways, and yeah, you need to know all 3. You can have a substitution, where one base is just swapped for another; an insertion, where an extra base is squeezed in; or a deletion, where a base is removed. A super simple way to remember them is with the acronym SID: substitution, insertion, deletion. Go ahead and write that down, SID. And now let's connect this right back to our case study. An exam question might ask you something like this: the change from DNA codon GAG to GTG causes sickle cell anemia. This is an example of what? Pause and think for a sec. The answer is substitution. That one single base swap from an A to a T changes the amino acid that gets made. That in turn alters the hemoglobin protein shape and causes the entire disease. This is a perfect, specific example to use in your answers. Okay, so while a substitution can be really serious, insertions and deletions, well, they're often catastrophic. Why? Because they can cause a frameshift mutation. This is a very high-yield concept, so listen up. Our DNA is read in 3-letter words called codons. If you insert or delete just one single letter, you shift the entire reading frame, and every single word from that point on is just completely misread. Let's use an analogy to make this crystal clear. Imagine your genetic code is this simple sentence: the big cat ate the rat. Your cell reads it in groups of 3, and it makes perfect sense. But now, look what happens if we just delete the letter B. The whole reading frame shifts. Now the cell reads the, then IGC, then ATA, then TET. It's complete nonsense from that point on. And the result? A completely non-functional protein. That is the key outcome you have to write in your exam answers. All right, we've totally mastered the single-letter typos. Now let's zoom out and look at the blueprint errors, those big, large-scale chromosomal mutations. We're moving on from single genes to these massive changes that affect entire chunks of chromosomes. So these blueprint errors come in 4 main flavors you need to memorize. There's deletion, where a whole segment is just lost; duplication, where it's repeated; inversion, where a segment gets flipped around; and translocation, where a piece breaks off and moves to a totally different chromosome. Unlike point mutations, these are massive structural changes that can affect hundreds of genes all at once. Okay, let's pause for a quick exam-style question. Which type of point mutation generally causes more damage? A substitution, our single typo, or an insertion or deletion that causes a frameshift? Think about that sentence analogy we just used. The answer is definitely the shifted frame. And for full marks, here's the reasoning you need to give. While a substitution can be harmful, it usually only messes up one amino acid. A frameshift mutation, on the other hand, alters every single amino acid after the mutation point. This almost always creates a completely useless protein. And that's why frameshift mutations are typically way more severe. So we know what these mutations are, but how do they happen? Are they just random mistakes, or can something actually cause them? This brings us to the agents of mutation. The key term here, the word you've got to know, is mutagens. Now, a mutagen isn't the mutation itself. It's the external factor, the chemical or the physical force that increases the rate of mutation. Getting that distinction right is absolutely essential for those tricky assertion reasoning questions. We basically group mutagens into two main categories that you have to know. First, you have physical mutagens, like X-rays and UV radiation. And then you have chemical mutagens, which are substances that directly mess with DNA, like mustard gas. Make sure you can name at least one example from each category for your exam. Here's a little specific detail that will make your answer stand out. UV light from the sun actually causes neighboring thymine bases in your DNA to stick together, and they form something called a thymine dimer. This creates a physical kink in the DNA, and it's a major source of errors when the cell tries to copy it. Mentioning this just shows a deeper level of understanding. Okay, so we've covered the types, we've covered the causes, and we've covered the effects. But now, why does any of this matter? Let's look at the big picture impact, which is going to help you answer those higher-level application questions. You know, understanding mutation is fundamental to all of modern biology. It's the raw material for evolution. That's a guaranteed link on your exam. It's the basis of cancer, which is really just a disease of accumulated mutations. It's how we diagnose genetic diseases, and it's how we develop treatments. This isn't just theory. This is the core of life-saving science. Okay, listen carefully, because this is a classic statement they love to use in true or false or multi-statement questions. Mutants make mutations more likely to happen, but they do not choose which mutation happens. The changes are random. So UV radiation increases your chance of a mutation, but it doesn't specifically aim to cause skin cancer. The damage is undirected. You have to remember that. So we'll end with this thought. On one hand, mutations cause terrible diseases, but on the other, they also provide the raw material for the evolution that created all of the amazing life on Earth. So are they a glitch, or are they a feature? It's something to think about. Okay, but before you go, let's do a super quick recap for your last-minute revision. Ready? Point mutations are side, substitution, insertion, deletion. Insertions and deletions cause frameshifts, and those are usually more severe. Mutagens increase the rate of mutation, but the changes are random. And sickle cell anemia is your go-to example for a substitution point mutation. I really hope this was helpful. Let me know down in the comments which part was the clearest for you. And hey, if you're serious about acing your biology exam, join our community of future toppers and subscribe. I'll see you in the next explainer.